Hello, this is Nagarajan, working as an assistant professor in Department of Electrical and Electronics Engineering, MAM School of Engineering, Trichy, accredited by NAC, approved by AICT New Delhi, affiliated to Anna University, Chennai. Okay. So today we are going to discuss about the one of the blood cell counting method techniques, that is hematocrit technique. Okay, hematocrit method. Okay, so what is this hematocrit, and why it has to be measured, and everything we are going to discuss now. Okay, so hematocrit method is commonly called as HCT in labs. Okay, HCT method. Okay, doctor will suggest HCT test. Okay, so HCT is the calculating the percentage of RBC in our blood, that is red blood corpuscles. It is also called as erythrocytes. Okay, erythrocytes in our blood. Okay, so it is given in the first point, that is to determine the RBC portion of blood cells in each volume of blood. Hematocrit or packed cell volume is used. Okay, hematocrit or packed cell volume is used. In the sense, hematocrit method or packed cell volume method is also used. It is all, that is, hematocrit is also called as packed cell volume method. Okay. So here what we are doing is, as blood samples is taken in a capillary tube. Okay, capillary tube is a small test tube like structure. Okay, it is very small in the sense it will look like a small pen like structure, very small test tube. Okay, and one end of the tube is sealed and with plastic material. Okay, one end of the tube is sealed with the plastic material, in the sense it is lidded. Okay, it is lidded completely. Then what it has to be done is, the tube is then rotated with the high speed centrifugal. Okay, with high speed centrifugal. Okay, that is usually we can see in let me show that one in the last slide. Okay, what is this centrifugal tub? Okay, it is usually rotated in the centrifugal tub. So what happens when it rotates? It rotates to separate the cells. Okay, to separate the cells from plasma. Okay, to separate the cells from plasma. So from the tube, the blood and cell volume can be compared by measuring the length of column. Okay, what is that length of column? It is very simple. There is a measuring stand. Okay, there will be a measuring stand. After this rotation is completed, that sealed test tube, capillary tube, is placed in that stand. Okay, that is marked stand. So there we can find, after the settle down, that is after the separation of the plasma and blood cells, we can find the two portions in that capillary tube. One is red in color and another one is yellowish in color. Okay, golden yellowish in color. So golden yellowish color is the plasma material, and red color is the blood cell materials. Okay, blood cell portions. Okay, so that what is that measured column. Okay, that measuring column, actually that measuring column is nothing but nanogram. Okay, it's nothing but nanogram, where we can take the readings directly by visualizing. So these red blood cells have high electrical conductivity than the blood plasma. Okay, Hence they settle down at the bottom of the capillary tube, and the hemoglobin concentration can be determined by destroying the members of the RBC. Okay, by destroying the members of the RBC, and then the hemoglobin is extracted and it is concentrated, concentration can be calculated. So next we will see the this method. Okay, so here you can see this is a capillary. Okay, this capillary filled with blood. After rotation we can see red color portion settle down. Okay, so this is the packed RBC, this is Hematocrit, and this is the plasma. Okay, this is the plasma, and this is a buffy where WBC and platelets will be there. Okay, so then this is placed in a measuring instrument, that is manometer. Then we can identify the amount of plasma, that is hematocrit present in our blood. So here is the small representation. This is the plasma. Okay, what is that? What plasma consists of water, proteins, nutrients, and here buffy is WBC and platelets, and this hematocrit is RBC. Okay, for normal blood, 37 to 47 percentage of hematocrit, and that is for male and female. For male it is 42 to 52 percentage. Okay, this is the normal one. This is the normal one, and for anemia. Okay, anemia patient, this RBC count will be very low. Okay, RBC count will be very low, and see, you can compare these two. Okay, here the anemia patient is having very low RBC and very more number of plasmas. Okay, for polycythemia. Okay, for polycythemia, the patient, here you can see RBC count is more, but plasma count is minimum. Okay, for this person the blood will be very light. Okay, the viscosity is very low. Okay, and for this person the blood the blood viscosity will be very high. Okay, it will be looking like when it is it will be very thick and it will be very light. That's all with this. And let me give small why this test has to be conducted. Okay, and here we can see. Okay, in case of high hematocrit, okay, high hematocrit in the sense polycythemia, it leads to heart disease, dehydration, and bone marrow disorders, and carbon monoxide poisoning. Okay, this leads to the patient. Okay, then testosterone use. These are the very tough situations, and if the hematocrit is low, it leads to blood loss. Okay, that is internal bleeding. Okay, then leukemia, that is bone marrow problem. Okay, and deficiency in iron and vitamins, and kidney disorder, thyroid. So these are the problems will occur when this hematocrit is low. So it is very important to maintain this hematocrit in the defined region, defined value, that is for male it is 42 to 52%, and for female it should be 37 to 47%. And here even for newborn babies it is very high. For newborn babies it is between 45 to 61 percentage, because they has to they has to produce their blood very fast, and it is very important to maintain this in this region. Hope you might have understood this hematocrit techniques, and remaining blood cell counting techniques we will see in the next class. So thank you.